last year, or year before last, I guess now, we had a paper in Nature where we took one of these populations and we sequenced a number of individuals from that population at from several different time points. So this has a lot of data on it, but I just sort of want to orient you. What we've got is we've got the sequence of the ancestral genome. Then we have a sequence of a genome from 2,000 generations, another genome from 5, another from 10, another from 15, and another from 20,000 generations. And we've put all of the mutations that we found up there. We see like this big orange bar. That's an inversion where about a third of the genome has had an inversion in it. But there are point mutations, deletions, all kinds of mutations. And in fact, at 20,000 generations, this particular individual in the population had 45 differences from it. So we wanted to distinguish between these two hypotheses. And so Zachary Blount, extremely talented graduate student in my lab, set out to do just this. This is about 1/3 of the Petri dishes that Zachary used in several experiments. What he did was, luckily there are not very many, as you'll become apparent in a second, there weren't very many colonies to count on most of these. What he essentially did was he took clones. So again, the question is not, was there a rare citrate positive variant? That's an interesting question, but he's saying, was there a change in the potential to evolve this new phenotype? So what he did was he took individual clones and restarted evolution. He took hundreds of different clones from, generation 2,000 and 5,000 and 10,000 and 15,000 and 20,000 and so on up to before it had evolved. He took these clones, grew up masses of their cells, and plated them. We did a little bit of it in liquid, kind of like what I showed before, but to scale up this experiment, he did it on Petri dishes where he would put, say, 1 billion cells of a particular clone on a Petri dish where the only carbon source that was available, there was no glucose, the only thing that was available was the citrate. And he was asking, had there been a change in the probability of evolving and making that transition? So in total, Zachary tested, now we're getting up to kind of physicist sort of numbers. He tested 40 trillion cells outside of probably things done in industry. I think this is the largest mutational screen I've certainly heard about being done in the biological realm. He had lots of controls in here involving the ancestral cells, so he actually tested about 10 trillion of the ancestral cells. Zippo. Not a single one of them ever was able to make this transition of using the citrate. So I said, well,